Hi everyone, welcome to my channel Pure Biology, and today I come with the continuing video that is related to GPCR and G protein. Okay, the activity of the GPCR and the G protein. Here I will try to describe. Okay, and obviously you just check out in my previous video where I already explained about the GPCR and the G proteins. Okay, so let's continue. Here the in G protein, there are three subunits. One is alpha, another is beta, and gamma. Okay, and the alpha subunit, that is a switch protein. Okay, and obviously GTPase switch protein. And the activity of the GTPase is what? Its activity is the hydrolyzation of GTP into GDP and PI. Okay, so this acts as the switch protein, and that alternates between an active state with bound GTP and obviously inactive state with bound GDP. Clear? So when GTP bound with the alpha, then it becomes inactive state, or it is in the form of inactive state. But when GTP is bound with the alpha, then it becomes active state. So when GDP is bound with alpha, it is inactive state, and when GTP is bound to alpha, that is the active state. So when G protein to an activated receptor, binding of G protein to an activated receptor, what is occurring here? That leads to the dissociation of GDP. When G protein that bound to the receptor, then the GDP that present in there, that is dissociated. And what is happening here? The G alpha is the G protein alpha, where the GDP has already bound, that is dissociated, and G alpha is bound with the GDP. Okay, and this when GDP concentration is very high, then G alpha is bound to the GDP, and when G alpha bound to GDP, as its GTPase activity is present in G alpha, so its G alpha try to hydrolyze this GTP into GDP and PI. And again, what situation is occurring here? That in the previous situation, alpha, beta, gamma both remain in attached form. Beta gamma in one form and alpha in another form. But when G alpha, that is bound with the GTP, it is totally dissociated from beta gamma. That means GTP. So here what is happening? Alpha GTP and beta gamma. Okay, this is actually occurring. Okay, so these two compounds, what is the role of these two compounds? These can interact with the effector proteins. Okay, so these two can interact with the effector protein, and this is happening in presence of obviously GTP. When GTP is there, GTP bound to alpha, it is separated from beta gamma. Clear? And totally the two, they activate the effector proteins, and these effector proteins leading to the changes their activity. So here it's actual role of this, the changing activity of the effector molecules. Clear? And the G alpha inactivates G alpha GTP by catalyzing GTP hydrolysis, and after that, after some time, GTP is activity of the G alpha. It is what? It's just hydrolyze the GTP and to form GDP and PI, and G alpha again bound to the GDP and dissociate from the effector. Clear? And reassociate with the G beta gamma. So what is happening in that situation? So when GDP and G alpha is formed, then it dissociate from the effector molecules, and G alpha again reassociate with the beta gamma. So the GTPase activity is of the G alpha that determines the length of the time that the signal remains on. Okay, so it is very important because it determines the length of that time that the signal remains on. Clear? So in some cases, RGS, the full form of RGS is that regulator of G protein signaling. This accelerates GTP hydrolysis, okay, by the G alpha subunits. So its function is that only the acceleration of the G alpha, nothing another work, okay, only the acceleration of the G alpha is the function of the RGS, okay. So RGS protein act as the alpha subunit specific GTPase activating protein, or you can say GAPS. The activation of an individual G protein may cause the activation or inhibition of the specific effector. So the activation of the G protein also dependent on the activation or the inhibition of the specific effector. Okay, so there are many effectors. One is the adenylate cyclase, second one is the phospholipase C beta, CGMP specific phosphodiesterase, and GEF of the monomeric G protein Rho, beta adrenergic receptor kinase, GIO regulated potassium channel voltage, controlled calcium channel, and PI3K kinase. Okay, so next, in some case where the GS is present, okay, then GS alpha compound is formed, okay, and this GS alpha activates the effector molecule, that is the adenylate cyclase. Okay, so GS alpha that activates the effector adenylate cyclase, and G protein involved in enzyme activation. Here G protein that is involved in enzyme activation. Okay, so here the enzyme activation is another role of the G protein. So that's why GS alpha first activate adenylate cyclase, and here the GS alpha binds to GDP and hydrolyze and activates the plasma membrane bound enzyme adenylate cyclase. Okay, and this adenylate cyclase that synthesizes the CAMP from ATP. So here S is the stimulatory G protein. S is the GS is the stimulatory G protein. And in the last case, what is saying that GS alpha that binds and hydrolyzes the GTP. Okay, GS alpha here bind with the GTP and activates the adenylate cyclase, and by the activation of this adenylate cyclase, the CAMP, synthesis of the CAMP is seen from ATP. Okay, so this is the working process of the GPCR and the G protein, and here the very short or the brief diagram, that is this is the plasma membrane and these are the GPCR or the receptors, and here the G protein alpha and beta gamma. Okay, these are in the sorry. Okay, so this is the GPCR receptor, and here the alpha beta gamma. Alpha here already is bound with the GTP, and beta gamma complex is separated from the GTP alpha, and here the adenylate cyclase that is activated, or this effector molecule is activated, and by the activation of this effector molecule CAMP generated. Okay, is generated. So this is the rough structure, and I will try to draw the activation of the GTP and G protein in the each part, but after wrapping this one. Okay. Okay, so this is the brief structure. This is the plasma membrane, and this is the GPCR, and this is the G protein. Okay, and this situation is the inactivated G protein. Clear? In the second step, when the signal molecules just attached with the GPCR, then what happening? The signal comes towards the G protein, and obviously in that situation GDP already there, so GDP is bound to the alpha, that means GDP alpha complex is there, so that's why alpha is already bound to with the beta gamma complex. But in the next step you can see that alpha is separated from the beta gamma complex because GTP is there in that situation, and that's why GTP is attached to alpha and form GTP alpha complex, and this is the activated alpha subunit, and now this is also activated alpha beta complex. Okay, so in that way GPCR and G protein both are activated. Clear? So this is the very rough structure. I think you can understood, and you just check out any book just after seeing the video, what is happening in the by the process, or what is happening due to the G protein and the GPCR when present in the plasma membrane. Clear? And signal is already enter into the plasma membrane. So what is happening, you can just see this video, and then you consult any book that is related to the cell biology, so it becomes clear to you. Okay, so thank you, and I will try to another topic in my next video, obviously related to the cell signal.